All right, today we are going to discuss chapter 37, and it's going to cover defenses against diseases, which for the most part will talk about the immune system. So here we have a case study over a flesh-eating bacteria. It's interesting because in, I think, the last chapter, we talked about how bacteria can negatively affect humans due to their production of toxins. It's interesting because we didn't really deep dive into that, but bacteria can have either endotoxins or exotoxins. Typically, when we think of toxins, we think of something secreted out of the cell into the surroundings. And so those would be called the exotoxins. And so it's both toxins, enzymes, and proteins, presumably needed for development or formation of a biofilm or adhering to a surface. Presumably, there's an actual reason why the bacteria are producing these toxins. It isn't simply to disable the host. However, it is to reproduce and to colonize, though. Um, so the idea is that from an evolutionary standpoint, the microbes invade us probably don't want to kill us because then they would lose the host. However, it does appear that some toxins released by bacteria don't really help it at all, other than to cause disease. So I guess that's still up for debate. But endotoxins, they're by far the most interesting. They're components of the actual bacteria that are toxic to humans that mediate an immune response. Um, Most often, if the bacteria is ruptured, then those internal components of it are exposed outside to the blood. So bacteria can be harmful both because, well, traditionally you would think of an endotoxin more so like a poison. So it's poisonous when exposed to, whereas exotoxins, they're actively secreted. And so you can think of those more so as like traditional toxins. There's a little bit of an interesting overlap when you look at the animal kingdom. So the difference between centipedes and millipedes, for instance, millipedes are poisonous, so the predators that ingest them will get sick and presumably die. However, so that's millipedes. For centipedes, though, they use venom and they actually will actively hunt and kill off their prey using a toxic venom. So there's a little bit of a difference between toxin and venom traditionally when we talk about animals. So I did want to specify that there are differences between the kinds of toxins, proteins, and enzymes that the bacteria produce. Endotoxin versus exotoxin. Ultimately, the question is how does the body defend itself from infection? And what does it defend itself against? Well, most of you probably know already it's going to be microbes. We've talked about that before. So microbes can be bacteria, protists, fungi, um, maybe technically you may want to classify viruses as microbes. They are microscopic, but viruses aren't living. But you also have potentially some parasites could potentially be microscopic, or at least start off microscopic, and or their larvae may be microscopic while they're reproducing. And so microbes that cause disease are going to be pathogenic, and by disease it means that they damage tissue or damage cells. So we did talk in the past about harmless bacteria, so or potentially beneficial bacteria. So the difference between, um, say, like, so there's symbiotic relationships. You can have one where one person benefits and the other isn't harmed, and then you can have another kind of symbiotic relationship where both parties benefit. So we have those two, both of those cases, as far as the microbes that colonize our skin and some of our uh, mucus passages like our nose. When it comes to protection against foreign invaders, and even we'll talk a little bit about internal issues as well. Um, you basically have non-specific uh, barriers, non-specific defenses, and specific defenses. So specific defenses target one specific thing or kind of thing at a time, basically. So we'll go into that a little bit later. But non-specific structures just block out essentially everything, or at least that's their goal. And so typically you think of the skin as that first line of defense. So if you cut your skin, you're at greater risk of infection than if you never cut your skin. You also have enzymes and proteins in tears, saliva, and your mucus that help protect against just general foreign invaders. So if you get invaded by, if that non-specific barrier gets penetrated, then potentially microbes could come into your bodily fluids or your tissues, and then you'll have that second line of defense, the innate immune response. And if that isn't fully sufficient, then your body will come with the adaptive immune response. And so the adaptive immune response, that's going to be the most targeted and specific kind of response. So barriers are important anytime you have external surfaces or cavities that open up into the external environment. So things like your mouth, which includes part of your mouth and your nose, so respiratory and digestive tract. So the innate immune response, that's going to encompass white blood cells, so part of your immune system that specializes in engulfing foreign particles, and they can be triggered by chemicals released. So kind of when you get swelling and inflammation, that's going to be um, the start of the innate immune response. And then the last or third line of defense is the adaptive immune response, in which case you're going to use specialized immune cells that will remember that kind of invader, so we'll have an easier time fighting it again. So innate, so the second and third responses, innate versus adaptive. The innate one is non-specific again, um, and it's a quicker response because it isn't trying to read or identify the particular kind of microbe and seeing um, specifically if you've encountered it before, and each time it encounters it, it will behave in the same way. Whereas the adaptive immune response takes a while to ramp up, and so um, during when you encounter it the first time, and then later on, uh, once it encounters the pathogen again, best case scenario, it's able to quickly ramp up that second time. Whereas the first time, it kind of has to do a little bit of a recognition dance, which we'll talk about the different cells that all coordinate together, immune cells that all coordinate together to sort of identify and remember these very specific invaders. And here you can see we have all sorts of kinds of immune cells. neutrophils, dendritic cells, macrophages, natural killers, mast cells, B cells, and T cells. And there are different kinds of B cells and T cells. And we'll talk a little bit about their roles later on in this lecture. So again, invertebrates, they only have the non-specific external barriers and non-specific internal defenses, whereas vertebrates uh, have the adaptive immune response. So if you've ever wondered why your nose is covered in mucus or your eyes are constantly well, that's weird. Of course, you expect your eyes to be wet. I mean, that's kind of like wet and lubricated. But why um, surfaces like your mouth, your nose, and your eyes are wet and moist has to do with mucus and different proteins and enzymes dissolved in that mucus to help fight off invaders. So even so, your skin it has a layer of dead cells um, that leave when you interact with everyday substances that fall off and are replaced. Um, so the like living your skin has multiple layers, and the outermost layer is going to be dead cells. So as those dead cells kind of fall or slough off, it's going to be replaced by a new layer of dry dead cells. And the idea is that the surface of your skin, because it's interacting with the external environment all the time, if you're constantly making a new layer, that kind of reduces the likelihood that harmful bacteria can establish a colony on you. And then your different proteins and fluids that lubricate or get secreted on your skin, like sweat and sebum from the sebaceous glands, in that sweat you can have natural antibiotics, for instance. And then mucus can physically trap microbes as well. It's actually interesting because from a like immune system response, the bacteria or fungi or pollen or whatever that gets trapped in your nose, it's kind of sort of beneficial for it to get swept back into your throat and you swallow it so that stomach acid can then take care of it. It's either that or you sneeze it out, but it's kind of funny that one of the easiest ways to take care of something that you breathe in is just to swallow it. So you actually have cilia that beat in rhythmic motion. They form sort of a current that flushes the what's trapped in the mucus down to your throat so you can swallow it. And that's called a mucociliary escalator. So continuing with the non-specific immune system, in there you're going to have white blood cells that are non-specific, this sort of patrol, and the inflammation response, like this redness and swelling, we'll talk a little bit about exactly what that is, and fever. They're all like normal parts of the non-specific immune response. All right, so white blood cells, they are formerly known as leukocytes, and there's quite a few kinds, such as phagocytes and natural killer cells. And so phagocytes, they actually physically eat. So phagocytosis, it's a form of cellular digestion, basically, where a cell will be able to intake something external to it, and incorporate it into the cell so it can then digest it. So macrophages, which literally mean big eaters, and then you have neutrophils and dendritic cells. They're going to be the most important kinds of phagocytes that we talk about today. So they consume both living and non-living foreign substances. So natural killer cells, they are unique because they can actually destroy cells. So bacteria can exist. Bacteria tends to colonize in your interstitial fluid or outside of cells, whereas viruses tend to colonize inside cells. So you have the macrophages, which will specialize in eating something in the bodily fluid, and then the natural killer cells trying to target infected cells to digest them. So if a cell gets infected, it's actually easier to just digest that cell and have the healthier cells replicate to fill in that missing gap rather than trying to enter into the cell and correct the error. You really don't quite see that. So how does the sort of immune system recognize a self from non-self? Turns out, so there's a sort of a process by which immune cells and later on more both non-specific and specific immune cells, they learn to, so in the process of development, they learn to recognize self versus non-self. However, self is identified by a whole set of proteins that are exists on the external surface of the cell. So the external surface of the cell is how the cell interacts with other cells and the external environment. Because cells don't have eyes or hands, so instead they rely on things like cellular receptors and carbohydrate tags and things like that, so that they can both be seen and see others, essentially. So collectively, these sets of proteins are called the major histocompatibility complex. And so that's going to be unique to you as a person. So your major histocompatibility complex can be thought of as you. So in the case of cancer cells, because cancer cells do not develop normally and they are abnormal, they don't look the same based off of those proteins on the outside. So they can be targeted by the immune system on the basis of like external like protein recognition. So say you cut your hand, um, and so your hands are what's used to interact with your external environment. You touch everything. I've talked about in past videos that the reason why your hands are dirty is because they're used to touch everything. And most things you interact with, you aren't washing on a regular basis. And so you cut your hand, you've reached that primary defense, your skin, and so you have to deal within the second and third lines of defense to prevent infection. And so part of that is going to be redness, tenderness, and so blood's going to rush to the area. Some of that will involve like clotting factors as well to seal up the wound. And then you're going to have immune cells rush there to um, to wall off the injured area as well as sort of ingest those foreign particles that do arrive there. So damaged cells, they're going to cause inflammation by releasing chemicals that trigger mast cells, and mast cells are going to release histamine. So if you have seasonal allergies, you've probably heard them referred to as antihistamines because antihistamines try to prohibit the degranulation of mast cells. So we talk about that a little bit later, but in order for blood to rush to an area, it has to change the permeability of the blood vessels. So snot and mucus are basically sort of your normal bodily fluid just leaking out of your your blood vessels so that so basically immune cells can also follow it in to the new site. So your nose is runny and your eyes are watery because your blood vessels are leaking fluid that is rich in immune cells to fight off the invaders. And so some kinds of white blood cells can actually squeeze through these capillaries and sort of get to the area as well. And so um, if you've ever seen like a part of your body that's damaged, cut, or scraped, and you can see sort of maybe like a white fluid there, and for some reason people are tempted to break open that sort of blister or vesicle and squeeze out the white stuff inside of it. Not only is that disgusting, but it's unnecessary, and you open up that skin back for potential infection. But it's there. That fluid is there because it's doing its job. In some cases, there's an excess amount, and it needs to be professionally lanced or cut open using sterile tools. Because if you just use non-sterile hands to pop an environment that was previously more or less sterilized, then you've reintroduced microbes back to the scene. A fever, again, that's non-specific. A fever isn't trying to kill one specific kind of microbe. It's trying to slow down the growth of all microbes that could possibly be in there. And so in order to have a fever, your body's normal temperature, its normal set point needs to be raised. So your normal body temperature is between 97 to 99 degrees Fahrenheit. There is some debate that normal body temperature is actually a little bit lower in the than it has been in the past, and some argue that that's because our ability to measure normal body temperature through like thermometers, increased accuracy of thermometers. So traditionally, you'll hear 98 point something, like 98.7 or 98.8, when in reality it's a little bit of a larger range. So it's interesting because if your set body temperature is supposed to be 98, but then a fever is what, anything above 100.2 or 100.4, a number really close around there, you're like, okay, well, what happened to those extra 2 degrees? Turns out there's a little bit of variability in your normal body temperature, and there's a threshold before it's considered a fever. Some doctors may say anything above 98 but less than 100 is a what is it? I think they called it a mild fever or something like that. But it's not considered a true fever. But I don't know. So in order to actually accomplish that, again, your thermostat, which is controlled by your hypothalamus is a portion of your brain, not to be confused with the thymus, which we'll talk about a little later. And in order to raise this temperature, there's the induction of pyrogens, which actually uh, everything in your body is done through communication through like chemicals that are released. So this is just one additional one. And it can also be used to reduce the concentration of iron in the blood. Iron's really important for invading microbes, one essential nutrient that they need. And so a lot of the iron we have in our body are bound up in forms that can't easily be used by microbes. And so that's on purpose. So that anything invades has to scavenge really hard to find that essential like nutrient. An elevated body temperature, it's going to increase the activity of your white blood cells while also slowing down bacterial cells. Um, those that are pathogenic have sort of evolved to reproduce best in normal body temperature. And so it can also help against viral infections by the triggering of an interferon protein, which will stimulate natural killer cells, which are, again, involved in dealing with infections inside of cells as opposed to infections like outside in the bodily fluid. That's also something interesting to note in case you hadn't sort of quite understood the whole picture. Even though your whole body is made out of cells, there are spaces between the cells. And so and you also have your blood, which has cells in it, like your red blood cells are in blood vessels, but there's still like empty space essentially in your body. Not every cell fits tight together like Lego bricks. So both that fluid between the cells, the interstitial fluid, and your blood fluid and your lymphatic fluid, which is a whole nother like vessel system in your body, that has to be patrolled as well. And so going back to fever, there can be too much of a good thing. Even with the fever too high, and it can, well, too high a fever, and you can start to denature proteins in your body. And the worst is proteins in your brain. And essentially, you can melt your brain with too high a temperature. But we kind of already know that. Whenever you cook your food, you cook it over fire, which is essentially an extreme form of heat. Obviously, a fever is not going to come anywhere close to the temperature of a fire. And even in an oven, you medium temperature is like 350 degrees Fahrenheit. Normal body temperature is 98 degrees Fahrenheit. So the adaptive immune response, that's going to be the one which we'll spend a little bit more time talking about. That's going to include macrophages, again, dendritic cells, and lymphocytes. And again, these are all kinds of leukocytes or white blood cells. And so the lymphocytes, they are specialized white blood cells that are specific to that third line of defense, that adaptive immune response. And so really you have your B cells and your T cells. It's interestingly enough that these grow and develop in the bone marrow. You can almost think for B cells for bone marrow. And so these immature immune cells start in the bone marrow. Some of them further develop and and become B cells. Other will leave and travel to the thymus and become T cells. And sort of this adaptive part of the immune cell system with these specialized B and T cells, they're going to actually have central hubs, essentially. Your bone marrow, which we just talked about, your lymphatic system, uh, specifically those nodules called the lymph nodes. When someone gets some sort of like infection, like a sore throat, one of the things that they may do is fill up at the top of their neck, just under their jaw, and there you have, it's quite common knowledge, you have two of the many lymph nodes you have. And when those are swollen, you're generally fighting off an infection. Your tonsils also, which are up there as well. And so your tonsils, if they get reoccurring like infections, you may actually be recommended that you get them removed. But of course, there's a disadvantage to removing some of those major hubs, because the tonsils are going to be that central hub for for your mouth, anything that enters via the mouth. So you have about 500 total scattered throughout. And the thymus, which I mentioned earlier, is above the heart. Um, and then your spleen, it's also involved in this as a hub, and it's on the left side of your abdomen. It's about fist size, which honestly kind of seems very large to me, unless I just have a really big hand. And so the spleen is going to be dealing both with filtering blood, breaking down old red blood cells, recycling them, and then uh, that filtered blood can be exposed to those leukocytes in its hub. And so some cells will secrete a certain kind of protein, and collectively they're called cytokines, and they're used for communication between cells, and they can simulate some of the white blood cells as part of their immune response and inflammation response. It's interesting because in the coronavirus pandemic, one thing you guys probably have heard about in the news, something called a cytokine storm. So it's talking about these proteins that are used to both communicate between cells and like induce inflammation and cell division among the white blood cells. So too much swelling can cause a problem, especially in certain areas of your body. For instance, your brain. You don't want it to swell because it's bounded by your skull, and all you're going to do is smash your brain up. Interestingly enough, side story, that's actually um, when your brain hits the inside of your skull, generally from like some sort of crash or accident. Um, That's actually a whole nother separate issue. It's generally not too fun. And so here we will talk a little bit about antibodies and the complement system. And so concussion, that's the term I was looking for, when your brain hits your skull in a result of an accident. So the complement system is super complex. We actually won't go too much into it, but they are mostly synthesized by the liver, and they circulate in your blood, and they assist the immune system. One issue that can potentially caused by certain kinds of microbes, ones that are, if you watch Monsters Inside of Me, they call them essentially suicide bombers, where the microbe essentially fragments into several pieces in its death, and each one of those pieces needs to be taken care of by the immune system. And so that there are so many tiny pieces that it leads to something called complement depletion, which can cause you issue with if you're trying to fight other infections that also work via complement proteins. So you quite literally run out of protein. And then antibodies, that's actually another thing that's pretty well talked about currently, especially during the coronavirus pandemic. You can get antibody testing to potentially see if you were exposed to the coronavirus and didn't get sick and got over it. Um, and so you could potentially be immune for a time period, not indefinitely though, or immune to a specific strain, and you have circulating defenses against it. Especially since not everyone has overt, like severe symptoms. The ones you hear about the most are the ones that have severe symptoms because You can be hospitalized due to your symptoms and have trouble breathing. But a whole group of people get such mild illnesses that they may not even realize that they're sick at all. And so they can be tested for um, circulating antibodies. And the antibodies would have been key to fighting off that infection. So in the adaptive immune response, you're going to have white blood cells recognizing an invading microbe as a foreign. Um, they'll launch their attack, and ideally, they will when they are able to eventually recognize it specifically. I mean, they're not going to know the genus and the species. They're only going to know it by its specific like antigen, which is a combination of basically proteins that identify it. So each pathogen is going to have a unique set of proteins. And so they're going to have a memory of what that unique set of proteins is and how to fight it off. And so that's what that memory is going to be of. So we've um, we've talked about antibodies a little bit, and we've talked about like pathogens. However, the part of the pathogen that triggers an immune response is called the antigen, and the antigen is short for antibody generating molecule. It's interesting because it's not only bacteria, but bacteria and fungi that have antigens, but actually viruses too, because once a virus enters a cell, well, it's kind of hidden from any part of the immune system that just patrols the blood. And so whenever a virus is active in a cell, it'll actually cause the protein tags on the outside of the cell to change, and new tags may get introduced, and some of those tags are going to be recognized as foreign because they are induced by the virus. They're not normally there, so if they aren't normally there, they're foreign. And so dendritic cells and macrophages, they engulf viruses or bacteria um, based off of their display of antigens. So they run across it and they're like, hey, this is not self, and so they're going to ingest it. And best case scenario, like break it up into fragments. And we'll talk a little bit about presenting as well later on what some of them do with those fragments. So lymphocytes, they produce two types of proteins um, that bind to specific antigens and trigger an immune response. So that's going to be antibodies, which are produced by B cells, and T cell receptors, which involve T cells. And so the big difference is B cells are going to produce basically antibodies, which is traditionally, we kind of think of everything as an antibody, but it's a specifically Y-shaped protein that binds to that antigen, that external protein, cellular protein that is presented, the what it's identified by, and it's going to bind to it, and that does a couple of things. It can either cause it to no longer be able to interact with another cell because its cellular receptor is covered, and it can also make it easier for other kinds of immune cells to ingest it and destroy it once its um, antigen receptors are covered. And so that, of course, deals with uh, pathogens that are in your bloodstream specifically. And then, okay, and then as far as the T cells go, it's a few slides. Here we go. The T cell receptors, Here we go. They are also going to be unique, just like B cells produce unique antibodies. Let's see. Okay, so we're going to go back to B cells. So B cells produce antibodies, and antibodies are also known as immunoglobulins. And there are five major categories of that: immunoglobulin M, D, G, A, and E. G is going to be the most abundant kind circulating in blood, like plasma. And E is also pretty interesting because you will, it's involved in allergies. So both food allergies and things like seasonal allergies are triggered the same way. And antibody, basically an immunoglobulin reacting to something that is harmless. But it's only the E type ones that are involved in this. Uh, Immunoglobulin G is also useful because it can cross the placenta and helps protect developing like fetuses or babies while they're in the womb. IgA, you're going to find that in surface digestive tract. It's covered in mucus, and the respiratory tract and saliva. And then A can also help developing babies whose immune systems have not properly developed yet. And here again is a structure of a B cell that has its unique antibody. And this antibody is going to match up with a protein on a microbe, the protein being specifically called the antigen or antibody producing or generating molecule. And T cells operate similarly, but they use a different kind of protein. It's not going to be shaped the same way. And they also bind to antigens. So both these B and T cells uh, have memory to them, and they both can divide to ramp up the amount. So because they each are unique, you need that specific one, like a lock and key model, to reproduce. So you have more to bind specifically to that system. And so B and T cells, as I mentioned, they are unique. They basically are randomly synthesized, and they contain, so you'll have antibodies circulating in your blood, potentially against diseases or pathogens or antigens that you haven't actually encountered yet or have encountered potentially in the past. So the difference is how abundant they are. If they run across the specific antigen, that specific kind will ramp up that has the right molecule to bind to the antigen. But you do have these antibodies circulating on a normal, or you do have these immunoglobulins circulating in a normal basis in your blood. And in fact, at any one time, you can have upwards of 100 million different antibodies, and then even more T cell receptors, which are basically the T version of immunoglobulins, because immunoglobulins are specifically generated by B cells. Interestingly enough, the immune system and And the B and T cells' ability to recognize self from non-self is the reason why you can't just, that's a weird way of phrasing, you can't just swap out organs between people. Organ transplants are often rejected. You have to take immunosuppressing drugs because the unique proteins are unique to the previous user of that organ. And your original, say, if you get I don't know, a heart transplant, it's going to have it's going to trigger an immune response because your immune system wasn't trained on the proteins of that other person's heart. And so regulatory T cells are important to help tamp down against autoimmune responses, so your immune system attacking itself. And also I mentioned earlier during their development, they basically get tested on whether or not they react to self, and ideally, if they do, they don't continue development. And they are marked for recycling. So because B cells and T cells are relatively, there's two categories of cell as opposed to all the other kinds of parts of the immune system that we talked about, macrophages, natural killer cells, neutrophils. Um, B and T are only a few of those kinds. Once they recognize an invader, they kind of, especially the first time, um, they kind of take a while to actually ramp up their response, and it can take um, between 1 and 2 weeks, especially that first time you see a microbe. And so once you run across it again, then it triggers that memory, in which case it ramps up production quicker that second time. But the first time, when it's your body is essentially just throwing keys at the lock, seeing what fits. As soon as it finds that right key finding it can be a few weeks. But after you know what key to use, next time you need to use that key, then it's a lot quicker. So we talked about the difference really between B cells and T cells. T cells mainly for dealing with infected cells, whereas B cells are dealing with the bodily fluid or the spaces between the cells. So humoral immunity, that's going to deal with the adaptive immune response in that bodily fluid between the cells, and cell-mediated immunity is going to be dealing with infected cells. And so, continuing with like antibodies, um, so the first step, circulating antibodies, they bind to a foreign molecule um, that can be a virus, and if it's then able to render it harmless through simply binding to it, it's called neutralization. So you're covering ideally the active sites. So this can be particularly useful in something that's, say, toxic, because a toxic substance no longer can interact if its active sites are covered. So like snake venom, um, that antitoxin is basically antibodies against that specific snake toxin. That's also why you need to know what kind of poisonous snake bit you, so you get the right antitoxin. And again, I mentioned it earlier, once antibodies have coated the surface of this antigen, it's easier for macrophages, which are the big eaters, and other phagocytes to destroy them. And here we just have a picture of that occurring. And this is where you get a little bit of the complement system interacting. The antibodies will interact with the complement system and punch holes in the plasma membrane of the microbe. So antibodies, again, they work in the fluid between the cells, and that's because they're really large and they can't actually cross into the cell itself. And so you'll see those working in the blood, the lymphatic fluid, and the interstitial fluid. And so viruses, again, they can hide inside of cells. Once they reproduce enough, they'll burst the cell and start circulating the bloodstream to find the next kind of cell. Well, the same kind of cell, but the next cell that's vulnerable to infection, because viruses specialize in a specific cell type. For instance, the cold and flu virus specialize in the cells that line, say, like your nose, different parts of that nasal pathway, and I think a little bit of the upper part of your throat. And so, um, flu is a little bit more destructive to those cells in the nose, which is why generally your symptoms last longer than the common cold. So, cell-mediated immunity dealing with the infected cells, and that's where you have your cytotoxic T cells coming in. And, oh, it also deals with cancer cells as well. So cytotoxic T cells can form pores in the infected cell's plasma membrane and can release enzymes into the infected cell, destroying it. And again, we've talked about how cancer cell often displays uh, proteins on the outside of it that aren't normal, therefore they're recognized as foreign, making it easy to fight them off. So turns out B cells and T cells can't operate just by themselves. They need the help of cleverly named helper T cells. So helper T cells also have T cell receptors, and these will bind to antigens displayed specifically on dendritic or macrophages that have engulfed foreign pathogens. So some of these cells will engulf the pathogen and destroy it, but take some of those fragments and put them on the outside of them. And then those outside fragments will then be bound by a helper T cell, which will then help a B cell or T cell recognize it and produce antibodies or the T cell equivalent to fight off that pathogen. And also these B and T cells develop memory by splitting it in two. Um, and some of them will form the plasma cell, which is actually making and secreting the antibodies, and then the memory cell, which contains sort of a copy of that antibody code, and a copy of that actual antibody protein. So these memory cells, memory B and memory T, can last quite a lot of time. Some diseases essentially last forever. Others a few years. And so the effectiveness also depends upon if the invader is sort of switching around what proteins it has on its outside surface. HIV is known to do this, which makes it really hard to come up with a vaccine against it because uh, it's not easily recognized. It's essentially a new novel thing whenever it changes those proteins on the outside of it. So the immune system only knows the previous configuration and can't fight off the new one. This is also one of the issues with the coronavirus pandemic, the SARS-CoV-2, the novel coronavirus, is mutating what's displayed on the outside. It's not a cell, but the outside of its structure. The antigen part of it is essentially slightly tweaked, and the B and T cells are specific enough that these slight tweaks may no longer work against it. As of this recording, there are now 3 strains or 3 variants of the virus, and each of those 3 are more easily spread. And so I believe there's the UK variant, and then there's I think it's a Europe variant, and then there's also a South African variant. And I think it's the South African variant that has developed mutations to make it more easily evade the immune system. Also, we don't talk about in this chapter, it's related to, I believe, our last one, microbes that have a slime coat around them. It's going to be difficult to actually have those antibodies directly interact with your cellular receptors if your cellular receptors are protected by a layer of slime. So certain microbes are harder to fight off because of that. You can't directly interact with the cellular surface. And so also the flu, the seasonal flu, it tends to mutate and change up those receptors on a frequent enough basis that the flu shot is a little bit, the mixture of what's in the vaccine is a little bit different. The antigens essentially in the vaccine are a little bit different combinations each year. And so some coronaviruses cause the common cold, and it's estimated that your immunity to the common cold coronaviruses to be about 6 months. You also have rhinoviruses, which cause cold viruses. So there isn't one cold virus. It's a whole classification that crosses different kinds of essentially species of viruses. And then MERS and SARS, um, Middle East Respiratory Syndrome and severe acute respiratory syndrome, they have between 24 and 36-month immunity once you get that infection. And the sort of hope is that the coronavirus, either you have lifelong immunity after you get infected, or you have greater than a year. Anything sub than a year, and all of a sudden you're going to run into problems with it, especially since the current vaccines require 2 doses weeks apart, and then you have weeks after that second dose before you get the full expected immune response. Imagine needing to take that every 6 months. You're basically taking 4 doses a year, everyone. You can see how that would be a problem. You want long lasting immunity. And so with the acquired immunity through interaction of those B and T cells and the formation of memory, once you first get exposed to it, it may take a few weeks to ramp up and find that right antibody to cancel out that antigen. And then over time, once the infection like wanes, that circulating antibody will be reduced. And if you come encounter it again, it will be able to ramp up a lot quicker and have a lot stronger response than when you initially saw it. So that's the principle behind vaccines, is that if you get exposed to the antigen or pathogen in a controlled setting, just a part of it, not the full thing, not enough to cause disease, your immune system will ramp up. And you can wait that extra few weeks for it to do so because it's not a real infection. And then when you find it in the wild for the first time, you can ramp up your response weeks quicker. So quickly that in fact, you may never actually get sick. You may never feel sick because the infection is taken care of before you even start to develop symptoms. It's kept in check, basically. So we have antimicrobials which can help you when your immune system is not properly equipped to fight off the infection. Antibiotics, they mainly deal with fighting off bacteria. You technically have antifungals for fungi because fungi are eukaryotic, so they're closer in structure to us human cells than bacteria are. Bacteria are prokaryotic and they're single-celled, whereas fungi can be multicellular and have specialized organelles. Bacteria are single-celled, don't have a nucleus, don't have membrane-bound organelles. So finding medicines to disrupt part of their replication are a little bit easier than fungi because anything that can disrupt their normal cellular processes probably will affect our normal cellular processes. And of course, your dosage and how long, so the quantity of antibiotic you have to take and the frequency and that full duration differs between what kind of infection it is. It's often common for you to be prescribed antibiotics for 2 full weeks. And that's generally a lot of time because if you miss dose, you reduce its efficacy. And that actually has to do with something called selective evolutionary pressure. We've talked about it in the past. In any population, by chance, you'll probably have um, microbes, so they are naturally resistant, but they have to compete for resources with the ones that aren't naturally resistant to an antibiotic. And as long as you aren't taking that antibiotic, then there isn't any preference between those two variants, and they both have to equally compete against each other. However, if you take an antibiotic and you don't take it long enough, you've killed off the competition for the resistant, drug resistant version, and it can grow unchecked then. So that's why you're never supposed to end your course of antibiotics early. Once you start to feel better, that doesn't mean anything. You got to keep taking your full course. And if you don't, well, you'll learn the hard way. You'll have to be prescribed a harder antibiotic that is either more specific or potentially, they're generally, the ones that are stronger generally have more side effects. And it's not going to be a really good time taking them because if you say you were prescribed 2 weeks and you finish after the first week, then you have to get a second dose because you cut the first dose early, and that second time is 2 or 3 weeks, and all of a sudden you have 1 to 2 weeks longer than if you finish your course correctly the first time. But even if you do everything correctly, drug-resistant strains can still occur, like MRSA, methicillin-resistant Staphylococcus aureus. So basically drug-resistant staph infections. Antivirals, they're interesting because for sort of a similar reason that we recently talked about is with antifungals is that viruses use your own cellular machinery against you. So anything that can interrupt a virus could potentially interrupt your normal cellular processes. Though with antifungals, because they have similar cell designs to humans, disrupting their cellular processes could disrupt our own. But in spite of that, we do still have some successful drugs to treat both fungi and viruses. For instance, you have the flu vaccine. It's not really treating it. It's a preventative measure, but you do have, I think, trademarked Tamiflu. It can actually reduce the longevity of your illness if you take it earlier enough in the infection cycle. And again, we talked about vaccines, allergies. They can be anything from food, mold, um, wasps, bee venom, milk, eggs, fish. They're all mediated by that immunoglobulin E. There is an interesting theory on that. It's part of the hygiene hypothesis. And so it's that if you're not exposed to enough microbes when you are young and like you're a child and you're developing your immune response, that IgE, that immunoglobulin E, is not going to be stimulated enough. It's the one actually responsible for uh, dealing primarily with parasites, so parasitic infections, like parasitic worms. And in, well, places like America, we're most likely not to run across them as much um, with our normal like hygiene standards and access to like you know, to clean products, to proper cooking, to waste that hasn't been contaminated, um, to water that hasn't been contaminated with waste. We're less likely to run up against parasitic infections. And of course, most times if you get one, it's because you've traveled outside the country or you've eaten raw or undercooked certain kinds of meat like pork. Which is known for having certain parasites. They can be killed when you properly cook them, but some people either don't understand or prefer the meat undercooked, or either it's a technical skill problem. Because we don't frequently run across that immunoglobulin E, that E antibody does those cells really don't get to exercise a lot, and they're hypersensitive to harmless things as a result. So this is definitely not an FDA approved treatment. However, some people have experimented with ingesting tapeworms to help get rid of their seasonal allergies. And you wonder why it's not approved. And they have to go to Mexico. It's the closest place they can get those. Okay, medical grade parasites, essentially, ones that you are purposely trying to ingest. So allergies are allergy causing antigens. They're called allergens. So antibodies, again, they attach to mast cells, mostly in the respiratory and digestive tract. So that's where you'll have your symptoms of eating a food allergy. Respiratory system, trouble breathing, closing of the throat, digestive system, water flooding into your colon, diluting your stool so that you essentially have diarrhea. Diarrhea induced by allergens. Or a milder case if you just breathe in the allergen, runny, stuffy nose. So degranulation of mast cells, rush and flooding of histamine, leading to leaky blood vessels, which cause mucus, mucus and other kinds of fluid to basically build up and form. And of course, your immune system can, in some rare cases, not properly develop, and it can either attack yourself, your own cells, and there aren't really good treatments for that, or it can fail to develop properly and not really fight off anything. So that would be an immunodeficiency problem. It can be triggered, or it can be genetic. Or it can be, yeah, genetic, and you can have it from birth. So it's congenital. So as a triggered, so radiation, chemotherapy, immunosuppressant drugs, or something you're born with, like diabetes, and then again something you can uh, basically some microbes can cause immunodeficiency. We talked about earlier, for instance, complement depletion. when the microbes explode and it uses up a whole set of immune cells trying to deal with every single piece or fragment. Congenital, again, something you're born with. Uh, Severe combined immunodeficiency can occur in newborns. And so, and something acquired later on in life, acquired immunodeficiency syndrome, AIDS, caused by HIV or the human immunodeficiency virus. And HIV mainly attacks helper T cells, which are needed to stimulate the antibody producing cells, the B cells, and the T cell equivalent, which deals with infections inside of cells. And it's cancer, uncontrolled replication of a body's own cells. So cancer cells tend to form tumors or masses of cells that grow unchecked and they don't develop normally in the sense that a normal cell responds to cellular checkpoints to make sure they properly replicate their DNA, to make sure they commit to dividing, things like that. Also, it's density-dependent inhibition. Cells, like skin cells, generally stop dividing once they touch. So cancer cells form tumors and lumps because they continue to divide even though they're touching. So we won't talk too much about cancer other than the fact that it can cause by damage to DNA in your cells and can also naturally rise due to errors in replication, and your immune system can sort of tidy up minor issues with just a few cells. Treating cancer, there's all sorts of novel approaches to that, but we really won't get into that. And of course, cancer can also be caused by uh, viruses as well, like HPV, which there's a vaccine out, and it causes HPV causes cervical cancer. All right, that about wraps up this chapter.